Hello everyone, my name is Esmeralda Rosas, and today I will be going over Chapter 14, The Natural Cycles of Vector-Borne Pathogens. So with this said, before I go on presenting, I wanted to go over some quick definitions. So just to cover, vector-borne diseases is defined as the illnesses that result from parasites, viruses, bacteria, or other pathogens that are transmitted by vectors. So some key components to know is the pathogen, vertebrate host, vector, and the environment. With this said, a pathogen is a bacteria, virus, or other microorganism that can cause disease, and whereas a vector is an insect or other arthropod, like a tick, that carries and transmits disease, causing pathogens, often called agents, to humans or other animals. So, ticks, mosquitoes, and fleas are some common vectors you might have already known about. And with this said, you want to keep in mind that just because a potential vector, like this tick, could carry a pathogen, it does not mean that they always will. So most of the time, they need to become infected first. And often a vector can become infected with a pathogen by feeding on another animal that is carrying the pathogen, and these animals are called reservoir hosts. So just to make clear, reservoir host is an animal that serves as a source of infection and can also sustain the pathogen in nature. So, having this said, some animals are actually better reservoir hosts than others. An example of this is white-footed mice and chipmunks are good reservoir hosts, specifically for the Lyme disease pathogen found in the northeastern United States. And to add on, other animals may get a pathogen if bitten by a vector, but can't transmit the pathogens back to another vector. And in this case, these are called dead-end hosts. So the white-tailed deer and animals are dead-end hosts for a pathogen that causes Lyme disease. So with all of this said, in summary, vector-borne diseases are those that are caused by a pathogen, transmitted by vectors, and often are maintained by reservoir hosts in the environment. The term arboviruses comes from the arthropod-borne viruses. So arthropods are things like insects, ticks, and they are vectors of these arthropod-borne viruses, meaning that they can transmit them. So some examples of arboviruses are the commonly known the flaviviruses, the yellow fever virus, West Nile virus, and Zika virus. One that I didn't list that's commonly known as dengue virus as well. And then we have alpha viruses consisting of chikungunya. And from the Bunya viruses, we have the Rift Valley fever and the lacrosse encephalitis, which was covered in the chapter. And so here I show you a arbovirus transmission model, and this is made up of a lot of questions that encompasses the virus, the vertebrate, and the vector. So the arbovirology would be this whole interconnectedness of the system that goes into transmission and how the transmission shapes up. So on the image below, the first one, we also have the added component of the environment. And I'll explain further on how it acts on this interaction of vector, the vertebrate, and the virus to sort of shape of how an epidemic might look like. So, having the arbovirus transmission system, when we cover epidemic shapes, we will see the shape like this image. So in this image, we have three randomly that generate the epidemics, and you can see one like the blue line. It starts in this beginning of this graph and has a long burn-in, and then turns into this epidemic. Or you have something like the yellow, which starts about the shorter burn-in but a higher peak, and it disappears, and it's also a little shorter. And then finally, you can have something like the explosive outbreaks. For example, one that is mostly known as chikungunya and Zika virus in the Americas, and that start out in the beginning as well and come all the way out to the top and then back, they die down. They come back down. So these are different shapes of what an epidemic might look like overall. And I thought this image was nice because the x-axis consists of time and y-axis might consist of number of cases, which can be applicable to any virus in a given population. So you might be asking, why is it important? Well, it's important to see how the dynamics of transmission are determined by the interactions of the transmission system, which there's an instance of like mosquito virus interaction with the vertebrate virus interaction and the environment virus interaction. So just to recap, something that is needed to know is what is a vector. So to refresh, a vector is something that transmits pathogens like viruses, and there are many types. There are many species that can be a vector. With this said, a main one commonly known as being mosquitoes, ticks, sandflies, and biting midges. And these are the biting midges responsible for blue tongue virus. So a tick can transmit several viruses, but also bacterial agents. And for instance, mosquitoes, they have many species of mosquito. There's actually, out of all of the many species, there is specifically 132 of the 80 species. And it's important to note that only the females bite. So mosquitoes, as a food source, they actually drink nectar, as you might have learned from other chapters, and so they are known as pollinators. So when females bite you, it's not because they're hungry, it's because they need the protein in the blood to then make eggs to reproduce. So ticks are also a long-lived arthropod, and with the ticks, both females and males can transmit viruses. So some vector considerations is I'll start with the vector competence. So this is often mathematically represented as a number that transmit divided by the number exposed. So one of the major things we study in vector-borne of arbovirus transmission is the vector competence. And this vector competence is actually defined as the intrinsic ability of a species vector to transmit a specific pathogen. And interestingly, not all mosquitoes that get exposed to a virus will become infectious. So we have this filter of time that determines this. So if I expose, for example, with this given graph, something you can reference with time on the x-axis and number of mosquitoes on the y-axis, you can determine this if you expose 100 of these mosquitoes. So it's possible of 60% of them will transmit a given a certain amount of time. So the time component is actually called the EIP, or extrinsic incubation period, and it's defined as the time between exposure of mosquito to an infection's blood meal to the mosquito is actually transmitting. So the duration of this incubation period actually depends on temperature as well as the species. So if you increase the temperature, it is known to shorten this extrinsic incubation period. And like I said, the extrinsic incubation period depends on the specific vector and virus involved. A typical extrinsic incubation period is around 10 to 14 days for the Bunia virus or flavivirus, and 6 to 7 days of an alpha virus. Moving on to the cycles component. So there is something called a sylvatic cycle. So a type of disease associated, this is a yellow fever virus, and this is maintained in the sylvatic cycle. So sylvatic actually derives from the definition of translates to jungle. So this is because it's mostly found in non-human primates and their association to the so-called jungle species, like for example mosquitoes in South America. So they actually maintain this cycle happily because it just keeps going, and it doesn't generally cause high mortality in non-human primates. So again, we call the non-human primates the reservoir host, and we call the associated sylvan vector the enzootic vector. So it's maintained in this big old sylvatic cycle. So as I covered, the reservoir host would be the non-human primates, and the enzootic vector would be the associated sylvan vector. So you might be wondering how or what happens, how does it get into a human outbreak? So what happens is, generally in this image you can see, it's what we call something called a spillover event. And that is when usually something we refer to as a bridge vector comes from the sylvatic cycle into an urban area and infects, if you want to think of it as patient zero, or somebody that's not infected in the urban cycle. So at that point, the yellow fever establishes, and sort of the independent urban cycle, where the bridge vector may or may not be the same vector, but it is not the same vector as in here. So those are two discrete cycles that are bridged by a bridge vector, essentially. So this little diagram can show us what may cause many different spillover effects given different factors. So with these many factors that I have listed here, that leads us to how, again, the environmental factors may impact diseases or disease outbreaks overall. So we have temperature effects. Like we said, it can change the extrinsic incubation period that can be affected. Different seasons may limit or enable when species come out. So for example, mosquitoes, for instance, they tend to be more active when it's in warmer climates, and they tend to not be as active when it gets colder. So things like that can affect how the viral replication rates and the vector biology overall, affecting all these different cycles that have been mentioned. So again, the vector population and the transmission dynamics. Now you may be wondering what is done with all of this components of viruses that exist. So there are some control strategies. This is an integrated vector management plan. So there are actually 4 categories of control tactics that can be used against all pests and vectors. We can think of how we see these tactics, or how we use these tactics, as a pyramid, with those that should be used, the hosts, at the base of the pyramid, and those that should be used the least, which are at the top. So overall, we can see that at the base of the pyramid are cultural controls, followed by the mechanical and physical controls, the biological controls, and lastly the chemical controls. So this is the same image, however, it has a little bit to it. So from the bottom to the top of the pyramid, we also see changes in the nature or the intervention, as well as the risk associated with them. So the tactics at the bottom, as you can see, tend to be preventative, while those towards the top require greater intervention. So as you go up this pyramid, the risk associated with using each tactic increases as well. So just to break it down, at the bottom we have cultural. So this cultural control is at the base of the pyramid, and these are behavioral changes actually that we can make to reduce the likelihood of coming into contact with such as we named, for instance, common are ticks or mosquitoes. So for instance, wearing long clothing to keep vectors out, or avoiding them by staying away from environments where you're likely to encounter them. This involves traveling as well. And then next we have the layer of the pyramid as mechanical control or physical control. And these are physical barriers that keep vectors away from you. So for instance, we have things such as window screens. So this keeps the mosquitoes out while you can still allow air in. And mechanical for ticks can include landscape modifications. So these are things such as fencing that actually reduce the number of hosts that may be carrying in your yard. So you can see this little deer that has a fence preventing it from entering. And another one is you could keep your grass short to reduce tick habitat. Following this pyramid, we have biological control as well. So these are living organisms that can be used to control ticks and mosquitoes, as we're using that as an example. So for example, there are mosquito fish which feed on aquatic mosquito larvae, and these may be added to ponds or other contained bodies of water to control mosquitoes. So this paper specifically discusses a tactic like this, the biological control. It's by Mataba et al. From 2021. And this article is basically saying how the predators present reduced different species that are found to be vectors. So they had backswimmers and dragonfly larvae to help the biological control component. And then we have the last tactic, which is the chemical control. So this may include any sort of synthetic or natural pesticide to control these ticks or mosquitoes, and they actually represent the highest level of intervention, if you remember that diagram, to the landscape, and the highest risks to the environment. So because of this, they should be used after careful consideration of the target vector, and they should be always used accordingly to the label instructions. And this is because you also want to have in mind how it can affect the biodiversity of where you are using them in. So all of these categories you want to look when designing an integrated vector management program. So the more you're aware of this, the more effective your approach may be. So with this said, I wanted to wrap up with some new research that has been researched since most recently compared to that of the book. So there are microvector interactions that have been researched on, so they have seen the role in disease transmission. So there is research focusing on how microbiomes within vectors influence pathogen transmission. So an example of this is that certain bacteria species within mosquitoes, for instance, can enhance or inhibit this viral replication, and this would impact the likelihood of the disease spreading. There has also been research where they have discovered a previously unknown vector pathogen interaction. So the one on your right, this is an article published in 2023, and so they found that this Colicoides peregrinus, it is a midge species. So they identified this to act as a potential vector for the leishmania, which is to expand the transmission. So with this said, there's also been advancements in vector control strategies. So you might have known from Dr. Vitek's presentation, he mentioned the vector control of Wolbachia infected mosquitoes. So this is used to suppress vector populations and to also decrease the pathogen transmission. So this has been seen to be pretty effective, and I kind of briefly put in you can see that picture is that actual midge species they discovered, so the potential vector, and to the right is actually how Wolbachia works. it basically affects the ability of disease transmission, or it can affect how the mating from it. So could produce like offspring that are not viable. So overall, the take-home messages is to understand the natural cycles that are important for effective disease control, and to also focus on control strategies such as surveillance methods and possible vaccine development. So we know from the past and from other research, there has been attempts for vaccine development. However, since all these mechanisms are so complex, and it's for some of the vaccines that have been developed are not so successful for everyone, or it doesn't work as a form to get rid of it, since as we know, viruses essentially have no cure. So with this said, that is all I have for today. I hope to see you guys for the in-person discussion. Thank you.